Good morning to everyone. I wish to my respect to everyone who attended this conference. Today I am going to try to present entitled Microalgae Products for Aquaculture. The section of this presentation consists of the species, their products, production system, and preservation as well. If you take a look at the contents in the first section, after a brief introduction about algae, I will give some common information about major culture species and about their usage area, and also production for aquaculture. And then I will talk about general nutritional properties such as protein, PFA, polyunsaturated fatty acid, and also pigments as well. And in the second section, I will talk a bit microalgae production system. For production of turbid larvae, which species are produced, and also what species are isolated from the Black Sea. Following this, shortly I will explain about production model made and Center Fisheries Research Institute. Lastly, I will mention about harvesting and then how to concentrate and its process and preservation of yield obtained. If some information about the general characteristic of algae will be, most of the algae are photosynthetic, like other high plants. Algae perform roughly 50% of the photosynthesis on this planet. Microalgae species are primary source of the animals in aquatic environment also. Algae represent a large and virtually unexplored group of organisms. Still there are 36,000 known species and more than 200 algae species worldwide. Over the last 4 decades, several hundred microalgae species have been tested as food, but probably less than 20 have gained widespread use in aquaculture. Most of the microalgae species produce unique products like carotenoids, antioxidants, fatty acids, enzymes, toxins, and sterol. The decline in the fish stocks and in the catch from wild fisheries in recent years has led to an ever-increasing focus on aquaculture, so they play a vital role in fish feeding. Major classes and genera of culture species, if you look at this, today more than 30 different species of microalgae isolated in different parts of the world are cultured at pure strains in intensive systems. You can see the table that the list of the major classes and 32 genera of cultured algae. Frequently used species in commercial mariculture operation are Cetaceros, Isochrysis, and Tetraselmis, Monochrysis, and Cruella species. The use of microalgae in aquaculture, if you look at this, the figure you see on the uses area of microalgae in aquaculture. As the primary producer in aquatic food chain, microalgae are the food source of many aquatic living organisms. So, unicellular marine algae are widely used as food in the hatchery production, commercially valuable fish and shellfish. Hatcheries rotifer cultured in just algae and then to themselves used as food for larva fish. Apart from this, in some systems are added to the water containing fish to improve the water quality. This model called as green water technique. About products, algae provides a variety of low-cost and rich source of protein and valuable components for human and animal consumption. Of course, it includes aquaculture sectors. In aquaculture, a few microalgae are used in the commercial applications. Species of Nanochloropsis and Tetraselmis generally used for feed applications, and as a result, produced algae powder and algae pastes products, also as extracts. The usage of microalgae pastes and powders being used generally in food and science area. Algae pastes are used as protein supplement in animal nutrition and aquaculture industry to feed larva fish, shrimps, bivalves, and so on. But the derived form, which includes powders and flakes, has various applications, extending from food, nutraceutical to feed industry. Okay, in this table, the comparison between algae paste and algae powder in storage conditions, under storage conditions. As you see in the table, algae powder water contents lesser than algae pastes. While algae paste are used in aquaculture as live feed, algae powder is used human health supplements and animal feed. Algae powder shelf life is longer than algae paste. And while algae paste storage in cold station, but powder in room temperature. Algae paste better digestibility and HUFA, highly unsaturated fatty acid, than about advantages. Unialgal cultures are essential when a high quality feed source with known nutritional properties is required. Bacteria-free algae cultures also reduce the risk of introducing unwanted pathogens into the animal cultures, which may result in mortality of some of the animals cultured. And live microalgae also inhibits bacterial growth, reduce overhead and production cost, leave food production space, and also easy to use and relatively long shelf life has. The most used species in aquaculture, if you look at this, a few different marine algae you see on such as Cetosaurus, Tetraselmis, Paradoxulum, Nanochloropsis. These are utilized commercially for making this kind of products. And apart from that, Spirulina and Chlorella are being used for a very long time, and these two have abundant proteins, especially beta-carotene, chlorophyll, antioxidants, minerals, and other essential nutrients, which are also processed into tablets. Similarly, Haematococcus and Dunaniella powder are used to extract bioactive components for pharmaceutical usage and cosmetic industry. Nutrition properties value of microalgae is influenced by their size, shape, digestibility, and biochemical composition. Several microalgae are considered to be as important feed ingredients containing high protein, carbohydrate, lipid, and antioxidant, and vitamin as well. In aquaculture practice, microalgae are important nutrition source of fish by direct consumption or as indirect profit. Lipids also are used at various growth stage of many aquaculture animals, including marine fish, larvae, bivalve, mollusks. Nutrition profile of some microalgae biomass, if you look at this, as you can see in the table, that protein, lipid, and carbohydrates rate of some species are very high. Protein maximum, you can see on table that 53% dry weight carbohydrate maximum can reach 50% of dry weight. Lipid also can reach maximum 69 of dry weight. And you can see in the last column of table that microalgae produced value-added components such as polyunsaturated fatty acids, pigments, essential amino acids as well. And as a last, I can say that microalgae species added to the culture time maintains the water quality as well. If you look at the proteins, these valuable components, proteins, as you had seen in before table, protein content of some algae species was high, and its productivity compared with soybean protein has much higher productivity, and making it this a feasible protein source for aquaculture. But, however, in some cases, due to palatability problems, microalgae feed may not perform well in aquaculture. So, palatability of microalgae feed should be comprehensively evaluated before use. The other one is polyunsaturated fatty acids, shortly PUFA. As you know that PUFA are not synthesized by the fish, but accumulated and stored from the microalgae. Compare it, you can see on the screen PUFA formation. Compare it with the traditional aquaculture feed, microalgae biomass contains much higher concentration of PUFAs. These fatty acids, such as EPA, DHA, linoleic acid, have been proven to be essential for the growth of larvae, especially essential at the early stage on marine fish larvae, and affects too much survival rate. Apart from this, what I can say, PUFAs affect spawning at quality and broodstock quality as well. EPA and DHA level is very important and are changed from the species to species of microalgae. As you see on figure that most microalgae species have from moderate to high protein, high percentage of EPA, changing from 7 to 34%. And non-necrolepsis has the highest percentage of arachidonic acid and EPA level. You can see on the figure. And Pavlova and Arthrochorus are relatively rich in DHA. As you see on figure that Dunanella and Chlorella are deficient in both EPA, DHA, and arachidonic acid. So these are not proper for larva fish. While the importance of polyunsaturated fatty acid is recognized, the quantity requirements of larval or juvenile animals feeding directly on microalgae should be well established before using. Let's move pigments. As you see on figure that Dunanella, the natural pigments such as astaxanthin, chlorophyll, and beta-carotene in microalgae are important to the growth of some fish species. The most well-known beta-carotene-rich microalgae is Dunanella, that has carotene around 14% of dry weight, and astaxanthin-rich microalgae is Haematococcus, that has astaxanthin around between 3% of dry weight. These pigments are used as a feed ingredients. So rainbow trout feed fat, astaxanthin, and have a better flesh color. And also some pigments play pivotal role in the immunity of aquatic animals. But microalgae species commonly used in aquaculture. In this table, you see the 12 microalgae species commonly used in aquaculture. Double plus shows the species that more popular ones than the one. If you take a look at the table, easily seen that the species are used to feed larval stage of bivalve, mollusk, abalone, crustacean, and zooplanktons enrichment and culture as well. In the second part, I will try to explain microalgae production systems, and then about the microalgae production model used in Central Fisheries Research Institute. General production is carried into parts. One in small-scale culture, starting from 10 milliliter test tube to 1 liter tanks. This process takes place until 5 liter culture in laboratory, and then from 100 liter to 1000 liter in acrylic tanks or polyethylene bags in greenhouse. The operation mode used here generally is a batch system, but semi-continuous system also. Other part is mass culture. These are usually operated in outside, using generally continuous mode or batch as well. For larger volumes, outdoor tanks and ponds are used, operated semi-continuously. And about the properties of microalgae to be produced, and microalgae species to be cultured must be an appropriate size for ingestion. For example, from 1 to 15 micrometer for filter feeders is necessary, like zooplanktons. They must have rapid growth rates and be stable in culture to any fluctuation in temperature, light, and nutrients as well, as may occur this situation in hatchery production systems. And finally, they must have a good nutrient composition, include an absence of toxins that might be transferred up to the food chain. And algae species can be easily obtained from culture collection or from their native nature by making isolation. And environmental demands of algae, what conditions is necessary. Algae are very sensitive to environmental factors. When it is appropriate nutrients, sufficient light and temperature grow very rapidly. On this slide, given some factors affecting the growth of microalgae. Now let's move for more information about these conditions. Light, each type of algae and optimum light intensity and a wavelength it has. In closed station like laboratory, light is normally provided by fluorescent lamps. The most commonly used types are cool white or daylight. Increasing the light intensity usually means better growth and faster division of algae cells, and it means production more biomass. But be careful, because lamps are generate heat and can make the culture too hot, especially in the closed place. You see in this slide the most common types on cool white or daylight bulbs. Light intensity and quality can be manipulated using with filters. Many microalgae species do not grow well under constant illumination. So a light-dark cycle is used. Usually 14 hours light and 10 hours or dark or 12 hours light, 12 hours dark is used. And about temperature at which cultures are maintained, it should ideally be as close as possible to the temperature at which the organisms were collected. Most type of algae grows well at temperatures from 17 to 22 degrees. A lower temperature will not usually kill the algae, but will reduce their growth rate. And above 27 degrees, most types of algae will not survive. Circulation is important to ensure optimum elimination of each algae cells. With adequate gas exchange, also effective possible temperature and pH control within the culture. The other one, nutrients. Seawater is a complex medium containing more than 15 known elements and a large and variable number of organic compounds. For algae culture, direct use of non-natural water is seldom accessible. Without addition of further nutrients and trace metals, the yield of algae is usually too low, and thus enrichment is normally required for algae cultures. Phosphorus is a limiting nutrient for algae growth. Algae can activate their growth features even when there is a phosphorus 20 milligram or under it. Silica is very important and necessary for diatoms, and in principle, the simplest comparison to make is whether the algae grow. Consult the culture collection websites for the most recent information on receipt and also to find the most appropriate mediums for a particular strain you will use. In practice, this is considerably complicated by the culture method. We use culture mediums for small scale culture and different mediums for large-scale cultures. We tried some culture mediums to check in that our culture propagate well or not. Mediums used were propolis enriched seawater, and Schreiber solution, and Valne medium. The result obtained that Valne medium was the most stable medium for small-scale culture of Nanocorapsis and for the other used for production. Medium for large scale, using micronutrients for large scale is very expensive, and also preparation is very difficult and takes too much time. So for large medium, only micronutrients are used and ensure well production of culture. This called as Yaşama medium, which composed of agriculture grades. You see on the formula on slide. This medium consists of nitrogen, phosphorus, and urea. You can see this formula on table. This standard medium is individually grinded in a mortar and boiled in a tap water for 20 minutes and applied to the culture tank in a rate of 1 milliliter to 1 liter. Other parameters: salinity of between 25 and 30 particle salinity units are generally best for the culture of flagellates, and between 20 and 25 particle salinity units for the culture of diatoms. These salinities can be obtained by diluting seawater with tap water if salinity high. We produce our culture in Black Sea salinity between 18 to 20 practical salinity units. About species from culture collection, in order to reduce labor and to avoid contamination between each culture, species, two species of phytoplankton, Nanocolepsis and Phaedoculum, have brought from the culture collection. You see the pics on slide, and much cultured in the first year of the project. You see the cell size, Nanocolepsis between 2 and 4 micrometer. Phaedoculum is more between 30 to 45 micrometer in size. Afterwards of the project, some algae species are isolated from the coast of Middle and eastern part of the Black Sea, and this is isolated species were identified by using molecular phylogenetic methods depending on the nucleotide sequences. You see the pics of these isolated species, and all isolates belongs to the Coleoptera division. These algae are Nanocolepsis oceanica, Chlorella vulgaris, Tetracelmis species, Scoccus deazoni, Cucumitsa parasitica. Primarily, studies on the production of these species in the cultural environment are still going on. You can see the production model in the table. Roughly, our production starts at laboratory in 10-meter volume test tubes and terminates outside in the tanks volume 8 to 30 ton tanks. Okay, now let's move to see the scale-up culture models. The process that starts in the test tubes continues in 200 milliliter of flask and ends with 5 liters of flask. Production takes 20 or 25 days under optimum conditions in laboratory. The test tube and flask should be shaken twice a day, but Balan Jauge starts to mix by the connection of air starting from this volume. The process that in greenhouse in the 100 liter acrylic tanks and finish 1000 liter tanks in the greenhouse. You see the preparation of the tanks before inoculation, and after preparation 5 liter cultured algae in Balan Jauge inoculate this 100 liter tanks and give Yashima medium 1 liter for 1000 milliliter and aerated well. Production takes 5 to 10 days under optimum conditions. The last part is mass culture. Mass culture is carried out in 4, 6, and 30 ton FRP tanks, which are located outdoor, sunny area, and tanks are spread from west to east position in order to promote algal growth for using sunlight. Vigorous aeration is supplied to agitate the algae culture well. You see on the screen the preparation of the aeration system on the peaks. Lastly, about harvesting, cultured algae that have reached their peak density are transferred to temperature control tanks before they are fed to rotifer or for concentration. Dense culture from highly productive ponds generally contain only approximately 0.1% of small algae biomass particles. Algae concentrate machine you see on. In order to minimize the manpower during seed production season and to have existing stock during off-season, and also as an emergency source for much culture. A part of all of harvested algae are concentrated through polyethylene hollow fiber membrane, 0.01 micrometer in opening diameter. You see on the slide a concentration process of phytoplankton. Approximately 1.5 ton of harvested algae are concentrated by machine and collected it in about 5 liters. Preservation, concentrated algae are preserved either in liquid or frozen form. This algae can be preserved more than a year without remarkable deterioration in quality. The concentrated algae are transferred into a plastic carboy provided with vigorous aeration and kept in the refrigerator at 1 to 3 degree temperature. The algae also can be packed in a plastic bag and preserved at below minus 30 degree in the freezer to prevent the oxidation of lipid content. Lastly, what I can say is the increase in aquaculture and more intensive aquaculture means an ever-increasing demand for microalgae. The present cost and variable quality of algae being produced significantly affect profitability of many aquaculture operations. In order to improve the yields and to reduce the cost, ongoing research and development is required on the discovery of new and better species of microalgae and cheaper and more reliable algae culture systems, and on ways to concentrate and preserve the algae biomass. Further opportunities for improving the economics of aquaculture lie in applying the experience of very large commercial procedures of algae, such as Chlorella spirina and Dunaniella, to microalgae production for aquaculture. Presently, microalgae are used worldwide as an alternative protein source, replacing fish meal successfully. In feeding trials with fish, many types of microalgae have been found to be used for increasing growth, feed utilization, psychological activity, stress response, starvation tolerance, disease resistance, and carcass quality. And although the algae kingdom is extraordinarily diverse and may comprise more than 200,000 species, Microalgal products at present are based on only 10-12 algae species, almost all of them microalgae. The development of culture systems for the production of microalgae, primarily in the Far East, has resulted in a stable supply of many of the microalgae of commercial value. Similar developments in the supply of microalgae are occurring today. Okay, that's all. Thank you very much for your invitation.